Here's a slide showing the genetics of flowering. You can divide the genetics of flowering conveniently into three stages. The first stage is flower initiation or activation of meristem identity genes. Once this happens, this leads to the activation of genes involved in regulating the spatial arrangement of the flower. Let's think of chairs being arranged in a meeting room. The final stage of flowering has to do as well who's going to sit in those chairs. In other words, where are the sepals and the petals and the stamens going to go, for example. In many plants, especially those that we call short day plants or long day plants, there is a factor called florigen that's produced in the leaves that actually activates flower initiation in the meristems. We now know this florigen is actually a protein called FT protein. In other plants, flowering can be triggered by the plant hormone gibberellin, and this occurs often in rosette type plants. Think of lettuce, cabbage. And still other plants, the plant size or age may actually produce factors that activate flowering. Interestingly, there's a protein called FLC protein that is present in the meristems of many plants, and this protein actually blocks flower initiation. In plants that need to overwinter, such as biennial plants, think carrots, winter wheat, beets, etc., there's a process called vernalization, which means the plant needs to go through a cold period before it can be competent to flower. What happens with vernalization is vernalization actually makes FLC protein degrade or go away, and this allows for subsequent flowering in the spring.